I'm delighted to be here because I so much appreciate the summer schools that Professor Cox and his team organize. I very much believe in the value of teaching and education, and I think that's very much needed in the context of mass spectrometry proteomics, making the methodology more broadly accessible. And this is one of the really excellent opportunities to spread the knowledge. And today I'm going to talk about single-cell proteomics. And usually I like starting with the motivation, why we would like to analyze proteins in single cells. It's difficult to do it, so we better have a good reason for that. And the reason that is oftentimes given is that our tissues are made of different cells. There is cell heterogeneity, and indeed that's an okay reason to do it, but cell heterogeneity can be analyzed by a variety of different methods. In particular, one can simply use optical microscopy or cell painting to analyze heterogeneity. What I think is special for single-cell mass spectrometry is the ability to use the methodology to ask questions about biological mechanisms, to be able to investigate causal relationships in protein networks that underlie the different functional biological behaviors that we observe. And mass spectrometry is particularly well suited for that because of its ability to measure proteins very specifically, as Judith Steen explained yesterday, the advantages over affinity reagents to detect epitopes. But of course, it's not just measuring abundances. We want to be able to measure enzyme activities, and we want to be able to measure synthesis and degradation rates. And this cartoon here from a recent Nature Methods paper that we published outlines some of the exciting applications that are enabled by single-cell protein measurements. And I'll talk about some of those, such as measuring proteolysis in single cells enabled by collaboration with the Cox team, and measuring and interpreting protein covariation. Briefly, I'll start at the beginning by outlining technology that is enabling single-cell protein analysis, and I'm going to focus on the big picture and the concepts that have motivated our efforts, what we have contributed to, and then I'll spend the majority of my time today sharing with you different vignettes of biological applications. To begin with, of course, we have to be concerned about sample preparation of very small cells. How do we prepare tiny cells without losing their proteins? Keeping proteins in, keeping contaminants out, that's clearly a challenge. And my group has worked on a number of different approaches for that, different generations. Here, I share two methods that we have selected because of their advantages. The first one uses multi-well plates, and cells could be sorted by a fax sorter. And this kind of arrangement is particularly accessible, and that's its advantage. Its downside is that we can only analyze, we can only prepare a few hundred cells at a time. And to use relatively large volumes, we also found that fax sorting is not particularly robust. We certainly had challenges with that. And with the ambition of overcoming those challenges and increasing the throughput, increasing the robustness, Andrew Leduc worked on a slide sample preparation method that in many ways is realizing my dream to begin with of how sample preparation should work. Before I had measured any proteins in single cells, I had started thinking about sample prep, and the dream was that we'll be able to do the sample prep on a glass slide, just a regular microscopic glass slide that doesn't have any patterning. We don't need expensive chips, just a simple glass slide where we can do the sample preparation in droplets. And Andrew Leduc, a doctoral student in my group, helped realize the dream where here, as you can see, we can lay out thousands of droplets on regular microscopic glass slides. And each of those droplets is going to serve as the reaction volume in which a single cell is going to be processed. And the flexibility of this design allows it easily for us to use it for both TMT Pro 18-plex or MTRAC 3-plex, or new plex DI reagents that have different plex. You can wake up in the morning, decide to do the experiment differently, program the instrument, and play out the droplets in a different design. And particular, I'm not going to go in details of for this method, but particular advantages to emphasize here, in addition to the high throughput, which we like, and the low cost, is the very low background that we see. We always worry a lot about contaminants contributing to the signal that we measure, because the signal that we measure, intensities, mass spec intensities, may not correspond only to single cells. They can also correspond to various contaminants. And this used to be a problem for us before we started using NPOP, and NPOP has been a very decisive step in terms of reducing contaminants in many ways, both background contaminants and +1 ions. So the first idea that helped us start doing single cell protein analysis was to use isobaric labeling to label peptides from small samples, such as single cells. And together we labeled peptides from 100 cells. And the idea here really is that what is difficult is peptide sequence identification. We need to detect sufficient number of fragments to determine the peptide sequence. And with this experimental design, the peptide fragments are going to be the pooled amount of peptides from all the single cells that are analyzed together and the carrier. And this really allowed to achieve higher level of robustness, higher depth of identification. And for the students in the audience, what I would also like to share as part of my personal story is that when we started doing this work, the community believed that mass spectrometry simply lacks the sensitivity to do single cell protein analysis. Very accomplished people with a lot of resources who have had the latest technology thought that this is not possible. One couldn't do single cell mass spectrometry proteomics. We did not have resources. We just had an idea that maybe it will work, maybe it would not work. We didn't know that. We did not have access to latest technology. We used relatively old instruments. So we did this 2016, used instruments from 2010. But it actually helped begin to change the thinking about what's feasible, because it challenged this notion that mass spectrometry is not sensitive enough. Of course, it wasn't easy to challenge the notion. There was a lot of skepticism. Does this really work? And I'll share with you just one snippet. I don't have it on these slides, but one snippet of data that I found very encouraging in those early days is that we could measure fluorescent proteins in single cells on a FACS sorter. And then we could also measure these same fluorescent proteins with mass spectrometry, and we found that the two measurements correlate strongly. And this evidence was quite convincing because we had two orthogonal measurements concurring on the same result. And as we were doing these results, we used MaxQuant a lot, and we took a lot of advantages, not only of the output peptide and protein abundances, but the extensive data tables that MaxQuant outputs, in particular the all peptides file. And what we noticed is that in experiments where we put a single cell label-free without doing anything too special, mass spec instruments done with reasonably careful chromatography, not even the latest mass spec instruments, can detect tens of thousands of peptide-like features. And I thought that was really noteworthy, because those peptide-like features, each of them corresponds to a peptide. Some of them are not identifiable. They might be modified, but they're present there at dozens of copies, some of them hundreds and thousands of copies. And we simply fail to assign confidence sequences to many of them. And this really framed the question for me very clearly, that the challenge is how to do the data interpretation. Very much a question that is very central to this summer school and to what you're discussing here. Can we come up with approaches to do this better? I think opportunities to do that still exist. And again, I want to emphasize this to students, that this is a young field that very much needs your bright, creative ideas. And I think that there is space to innovate, even if you don't have the most resources. You can try something else, new and different, that those with resources have not thought of. And our approaches to that were two complementary strategies. One of those is, well, if we detect tens of thousands, hundreds of thousands of peptide-like features, we don't have the time to analyze each one of those one at a time separately. But at least we can pick and choose. If we use real-time instrument control, we can tell the instrument to consistently select peptide-like features that are going to be identifiable, that are going to correspond to peptides of interest, and that are going to be useful for the analysis. And this gave rise to this prioritized analysis that we developed in collaboration with Christoph Wichmann and Jürgen Cox. I'll tell you a little bit more about it. And the other really complementary approach to that is, let's not isolate peptide-like features one at a time, right? If we don't have time for 60,000 peptide-like features, isolate it one at a time with long accumulation times, we can start isolating hundreds of them, hundreds of precursors, fragment them together. And of course, the challenge in that case was, can we do this in a multiplex fashion? And we worked towards developing PLEX-DA, and I'll also share with you some of the novelty there and what I think was important. But first, let me talk about prioritization. Prioritization very much built on existing strong framework that previously Christoph and Jürgen have developed of real-time instrument control using MaxQuant Live. But we had a challenge with MaxQuant Live because I wanted to go for very deep coverage. I wanted the instrument to be active all the time in doing MS2 scans. But if we did that, there was a risk that some of the precursors from the inclusion list will never be analyzed, and we were getting relatively low data completeness. So we could fluctuate between having high data completeness with a short inclusion list and therefore low depth of analysis, or we could have deeper coverage but with low data completeness. So to overcome that challenge, we came up with this prioritization scheme where a subset of precursors are going to be given high priority, and the instrument is going to analyze those first. And then if it has time, it's going to go to the middle, low priority tiers and others. And here this illustrates the logic. And we talked with Christoph, and he implemented this in MaxQuant Live for us. So let me show you the results of doing that. So first, looking at the motivation for doing it, we wanted to increase data completeness when we have maximally long inclusion list. And here the data, the black bars correspond to MaxQuant Live being run with a long inclusion list and no priority tiers. And just as you predict, we see that data completeness is not great simply because the instrument doesn't have time for everything on the inclusion list. It is detected, but sometimes we simply run out of time and precursors are not analyzed. When we introduce priorities here with the red bars, you can see the data completeness is much, much higher now for a list of thousands of prioritized peptides. And this doesn't come at the expense of lower protein coverage. If you look at the middle panel with the red box plot, you see that we actually quantify more proteins per run. And here are the details to illustrate that what Christoph did is perfectly implementing the logic that we wanted to implement. So you can see the number of detected precursors at MS1 level. We detect most of them at the high priority tier. Virtually all precursors are analyzed, 99% plus. At the medium tier, only about 60% are analyzed. At the low tier, only about 25%. And this is fine because the instrument has finite time. We cannot analyze all of them, but we can pick and choose. And then MaxQuant Live has robust and good enough instrument control in real time to perform the data acquisition that we wanted to perform. And because we select to fragment precursors that we know are identifiable, we see that we are able to assign high confidence sequences to the vast majority of MS2 scans. So we are not wasting precious instrument time fragmenting precursors that cannot be identified. And this results in doubling the number of peptides that can be identified and the number of proteins quantified per single cell. And again, we're using exactly the same hardware, exactly the same sample preparation. The only thing is that we use better real-time instrument control. And this also substantially increases the dynamic range of precursors that we can quantify. We are able to quantify less abundant proteins, including post-translationally modified peptides, as I'm going to show you later on. And we are doing this as we're doing it because we had such a good control of and good estimation of retention times in real time. We were able to sample elution peaks close to their apices, and this reduced co-isolation. So together with using very narrow isolation windows, we had a chance of achieving good quantification. But having a chance of good quantification is not the same as knowing that it's good. So we wanted to benchmark it, and frankly, reviewers encouraged us to do that gently as well. So we spiked in peptides, and this is done with the sample preparation that I talked about, NPOP by Andrew Leduc. He helped incorporate this easily into the sample prep. We spiked in peptides, synthetic peptides, at abundances comparable to the abundances of endogenous proteins. And then we compared the spiked in levels to the measured peptide levels, and we found that the agreement was remarkably good. And again, I attribute this to being able to minimize co-isolation. We cannot eliminate it. Co-isolation is there. It affects quantification. But if we sample close to the elution peak apex and we have a small enough isolation window, we see that quantification can be quite accurate. And now I'm shifting gears to talk about the other strategy, the other technology that looks at isolating in parallel many precursors, but we wanted to multiplex this so that we can analyze multiple cells at the same time. And multiplexing plex DIA is something that people had done previously. But in previous examples, when people had done it, the identification rates for peptides had decreased substantially. And if one is able to increase throughput, analyze more cells, but get 2 times or 3 times shallower proteome depth of coverage, that's not necessarily a good trade-off. There is a steep penalty to be paid for that. And our hypothesis and objective here was, can we multiplex the samples, but without paying a penalty price for that, without decreasing the depth of proteome coverage? And then the idea was we're going to test it with a 3-plex because those are the mass tags that we had at the time. But we really hope that this is a more general framework that then we can make non-isobaric mass tags to multiplex to much higher level, especially for single cells. So we think that the capacity to do that is there. And here I just emphasize with this approach again that we want to increase throughput, but very much conditioned on preserving the depth and the accuracy. We don't just want more data points. We want data points that are as accurate as what we can do with label-free analysis. And we want to maximize the delivery of ions to instruments. So when we analyze tiny samples, such as single cells, counting ions is very important. We always keep our eye on how many counts were sampled, because if we don't count enough ions, our statistics are very flimsy and quantification is not reliable. We are subject to counting noise. So with this frame, and this is well published, I'm not going to go through all the data from PLEX-DA that was published in Nature Biotechnology last year. I want to share with you some of our thinking for that and some of the advantages in more depth. And one of those is that if one uses certain mass tags, in particular mass tags that are not deuterated, some labels such as dimethyl labeling introduces deuterium that can shift retention times, but with certain mass tags, and in this case we're showing this data for Amtrak, retention times don't change for peptides labeled with different versions of the mass tags. So we could actually quantify how precisely the same peptide from different samples is going to coalute together. And when Jason Dirks did that, we saw that the retention time errors were very, very small within a run, much smaller than what we see with the best alignment possible between runs. And this retention time precision within a run gives us a lot of statistical power to be able to consistently detect precursors and fragments between different channels. And essentially, the higher precision, the higher accuracy is present both at the precursor level and the fragments. And at each of that level, it gives us more statistical power to robustly identify signal. And as Jorgen already explained this morning, how those features are being incorporated in MaxDIA, which is wonderful to see, and how this can be extended to isotopologous carriers. For us, this really has been a motivation from the beginning in developing PlexDIA, something analogous to what we did with the isobaric carrier. It is different in many ways, but in some ways it's analogous, that robust identifications can be achieved and enhanced by using a carrier sample combined with labeling. So we have been working on this to be done with data-independent acquisition in conjunction. And we have encountered challenges of being able to do it in a robust way and to not undermine the quantitative accuracy. It's very easy to introduce a carrier and increase the depth of protein coverage. In some ways, that's a strong expectation, but it also has the potential to undermine quantitative accuracy, as indeed some of the published examples show. So we used mixed species benchmarking and Luke Curry actually did those experiments to try to develop data acquisition schemes and analysis schemes to preserve quantitative accuracy. And here I show one example of such experiment where Luke used a carrier sample that was labeled with the heaviest label, delta 8 of MTRAC. And that increased protein coverage about twofold while preserving the quantitative accuracy. And Luke is working on writing a paper that is going to share the details on that, and we are going to introduce what are the important guidelines and things to observe when using such carriers so that the accuracy is preserved. But in the meantime, before we have published that, I would just point out that many of the same considerations that we previously shared for using isobaric carriers apply in this case of using isotopologous carriers as well. And one has to be mindful that the windows are going to be wider for isotopologous carrier with DA compared to what they are for DDA with isobaric carriers. And another direction for us with extending data-independent acquisition, of course, is taking advantage of trapped ion mobility spectrometry. And I spoke previously about the important observation that even on older instruments, we detected tens of thousands of peptide-like features. Now on timsTOF instruments, we detect even more. So we are very, very good at detecting them. But in our first instance of using timsTOF instruments with PlexDIA, we had 3 MS2 frames. And 3 MS2 frames means that only about a third of the ions are being used at MS2 level. And that means that the other 2/3 are being lost as we isolate. That's not bad. There are strategies to throw away more of the ions. But we wanted to see, can we improve that? Can we use larger fraction of the available ions? Because when we analyze tiny single cells, every ion can be helpful. And we have been thinking that one way of doing that is to use the fact that each slice in ion mobility space also corresponds to a slice in M/Z range. And then in principle, we can use this correspondence to have just one MS1 frame, one MS2 frame, and deconvolve the data. And the advantage of doing it this way is that now we can shorten our duty cycle by a factor of 2. We can more frequently sample elution peaks. We can have high probability of sampling close to the elution peak apex. And then we get all of the ions. We're not throwing away ions with this data acquisition. Of course, data interpretation is more challenging, but Vadim Dimitriev worked on incorporating a module in Dayan for interpreting this data. And I can show you some preliminary results from this acquired by Jason Dirks in my group. And what you can see here with this strategy is that because our duty cycle is only 200 milliseconds, even when we have a couple of second dilution peak, we can get dozens of samples of both precursors and peptide fragments. And this high frequency of sampling actually gives us more statistical power to associate precursors with the corresponding fragments. And then we see that also empirically, there is a substantial benefit of having the very frequent sampling at MS1 level. So this is a technical experiment in which we show that if we keep injecting exactly the same standard, but we decrease the number of the frequency at which we sample, in this case, just add more MS2 scans, but if we decrease the frequency of survey scans at MS1 level, we measure lower and lower precursor intensities. And of course, that is simply reflecting the fact that we have lower probability of sampling the apices. So higher frequency of sampling is certainly good with shorter duty cycle times. And we also see that when we go to very small cells that have less protein, this strategy of having one MS1 frame, one MS2 frame really pays off, as we also at the level of precursor identification, as we're able to identify more peptides. And I think that this is preliminary data to share. This is not closing the book on taking advantage of ion mobility and coming up with different data acquisition strategies, and in particular data interpretation strategies. I think there are a lot of opportunities for clever young people to innovate and contribute to this area. Good. So that's the technology that I wanted to share. And now I'm going to switch gears and talk more about the biology. What are we learning? And the first problem that I'll tell you about is about early mammalian development. Once upon a time, each and every one of you was a zygote. And then that zygote split into 2 cells, and then 2 cells became 4 cells. And a question that emerges here is, when is the symmetry broken? So what's interesting is that there are some measurements, functional measurements, to suggest that very early at 2-cell stage, the 2 sister blastomeres are functionally different. But it has been very difficult to identify molecular covariates of those differences. Indeed, if you measure messenger RNAs in those single cells, as many groups have done, you don't find many differences. A couple of messenger RNAs are maybe different, but it's not particularly compelling difference. So when we measured protein abundances at the 2-cell stage level, we found that there are hundreds of proteins that are differentially abundant between the sister blastomeres, and they're differentially abundant in a way that from the early to consistently we could identify one of the blastomeres as being alpha and the other beta, and those differences actually increase from early to late 2-cell stage. So that's interesting, but of course the question is, do those differences matter? Are they just molecular differences, or do they reflect functional, biologically meaningful specialization? So to answer this question, we took those 2-cell stage embryos, we cleaved them in 2 parts to remove the sister blastomeres, and one of them was analyzed by mass spectrometry to determine its type. And the other one was allowed to develop into a blastocyst, and then that blastocyst was assayed to be able to determine the cells from the different lineages and the health of the blastocyst. And the results from these experiments indicated that beta cells are far more likely to give rise to healthy embryos, to have a large number of epiblast cells. And this indicated that indeed those protein differences that we measure are indicative of biological developmental potential. Now, these data that I showed you initially came from mice, but of course we were interested how relevant are they to humans. So we were able to do some of those measurements also with human embryos. And we found that the differences are highly conserved, that we see the same proteins being different in human embryos. And the enrichment of biological functions is suggesting that what is driving that first breakage of symmetry is protein transport and protein degradation. And that provides a compelling explanation as to why those differences are not observed at the RNA level, because at that point, the genome is actually not very active. This is happening prior to the zygotic genome activation. What we have is protein remodeling by degradation and transport, beautifully illustrating the difference between measuring RNA and protein abundances. Now I'm going to switch gears and share with you a few studies that all looked at the possibility of interpreting protein covariation. So if we measure proteins across many single cells, we can look at that covariation and we can use it to discriminate between different classes of models, ideally, if we have highly accurate data. But in more modest examples, we can use it to just develop hypotheses and to test different ideas. So one such system where we have applied covariation analysis is epithelial to mesenchyme transition. It's a natural process that happens during development, and it's a very important process that is required for metastasis, cancer metastasis, and this makes it one of the most studied processes in biomedical research. And a hallmark of this process is that we have adhesion epithelial cells turning into motile mesenchymal cells, and this involves rearranging of their cytoskeletal proteins. And here is a very interesting observation that we made for that process. We observed that proteins of the cytoskeletal apparatus don't change in abundance in the course of EMT, but they change in covariation. So here we start with two proteins that are negatively correlated. And then as the cells differentiate, they become decorrelated, and then they go to positively correlated. So that's an example of a relationship that in principle we cannot observe from bulk analysis. But from single cell analysis, we are able to detect these changes in covariation. Exactly what it means, we are still figuring out. I cannot tell you, but I think it's pretty cool. So that's why I decided to share it. Another example comes from the work of Andrew Leduc, where we looked at melanoma cells that are where we have two clusters, and one of those clusters is becoming primed for drug resistance. And what is interesting is that within a cluster, which here is denoted by color, we can see that there is a strong covariation of proteins, which is suggesting that the single cells within that cluster are not just all identical or differing from each other in some noisy way because of stochastic fluctuations, but it's suggesting that there is a systematic signal. And in this particular case, Andrew was able to interpret that this signal really corresponds in some cases to functionally concerted sets of proteins that covariate together, such as glycolysis and oxidative phosphorylation. So what is happening with this gradient is that we see that some of these melanoma cells are relying more on glycolysis, likely for producing energy, while others are relying more on oxidative phosphorylation as they become more inclined to be primed for drug resistance. We can also look at covariation between RNAs and proteins. This is an early example from the work of Harris and Speck, where we were able to align measurements by single-cell RNA sequencing and by mass spectrometry. And here you see an example for a gene where the products of the gene are strongly correlated, suggesting that likely there isn't much post-transcriptional regulation. And an example to the opposite is P53, where we see no significant covariation between RNA and protein, likely because in this case we know that messenger RNA is constitutively transcribed, while most of the regulation of protein abundance occurs via protein degradation. And furthermore, we see that the protein abundances are indicative of the transcription factor activity, while the corresponding RNA template is not so. But what about this case? So here we see some weak covariation, and potentially there could be post-transcriptional regulation, or maybe there is a lot of measurement noise. And in fact, if you look at the RNA level, you can see those vertical stripes correspond to counting one messenger RNA per cell and two messenger RNAs, and that's associated with a lot of noise. So is there post-transcriptional regulation here? I don't know. I don't know. The only way to know is to be able to accurately benchmark how much is the noise so that we can say how much variation is expected because of the measurement noise, and then we can discount that and say if indeed there is post-transcriptional regulation. And we have been thinking of ways of doing that and have actually started developing approaches to do it, where we want to use orthogonal measurements so that we look at the covariation that is attributable to measurement type or ensemble and discount that from further analysis. And here is one example of doing that, where we compared measurements done by two very different methods on biological replicates, cells that were grown apart months apart of different cultures. And in one case, they were measured using prioritized scope, which I introduced, and that uses reporterines for quantification, or they were measured by plex DA, where the quantification is based on precursors, two very different methods. But nonetheless, they identify largely the same clusters. And even further, we can be more quantitative. We can compare directly the fold changes of proteins between these clusters, and we observe that the correlation is reasonably good, suggesting that a large fraction of the variance in the data correspond to biological signal, since whatever artifacts are present in one method are unlikely to be present in the other method. For example, co-isolation affecting prioritized scope is not going to affect plex DA in the same way. So it's good to see that the variance is dominated by biological signal. And another biological system that I'll share with you is macrophage polarization. A very simple model system where we looked at how primary macrophages derived from bone marrow cells react to LPS treatment. It's a commonly used biological paradigm to study response to inflammatory stimuli. And here you can see two example responses for two different functional groups of proteins. One is type 1 interferon signaling. And we can see that in the non-stimulated cells, the proteins are uniformly low. In the stimulated macrophages, they're uniformly high. And you didn't need single cell protein measurements for that. That's sort of the textbook expectation. Those cells are similar, they react in the same way. But if we look at phagosome maturation, we see exactly the opposite. We see that the differences are not between treatment categories, but they're within a treatment group. And that's interesting. But then again, we have the question, is this just molecular noise, or is this relevant to the functions of the cells? And in this case, we had a relatively straightforward way to test for the phagocytic activity of the cells by directly measuring it, by introducing fluorescently labeled dextran molecules and measuring the uptake efficiency. And we see that there is a 30-fold variation in the uptake efficiency within a treatment group, which is consistent with the variability that we see at the protein level. There is also functional variability. And not only that we can associate many proteins that are significantly correlated to that functional variability, but we can relate it back to the single cell protein variability that we measured and say that that gradient that we saw in single cells is related to the ability of cells to take up dextran. And furthermore, with this prioritized approach that we developed in collaboration with the Cox group, with Christoph, we are able now to prioritize proteolytically cleaved peptides, and one could use this for other post-translationally modified peptides. And we first validated this using standard assays for measuring proteolytically cleaved proteins. In bulk, we measured in single cells, validated that it works, and then we applied it to the single cells to measure those proteolytically cleaved peptides, reporting on the activity of caspases. And I think that this approach in general represents a promising strategy for studying post-translational modifications in single cells with mass spectrometry. How am I doing on time?
You have 15 minutes.
Okay.
Also, you can continue more if you have.
Oh, I would rather be shorter than longer, but I think that's plenty of time. So, okay. I have time to tell you about another project then, which is studying the emergence of drug priming in melanoma cells. And here I show an image from a very nice paper from Arjun Raju's laboratory, where they studied a particular melanoma cells in which they observed that there's this coincidence of jackpot cells that have very large number of messenger RNAs from some genes. And those cells where that coincidence is observed, it occurs with much, much higher probability than expected by chance, actually are primed to become drug resistant. And Andrew used this system to analyze at the protein level, and this was the first system to which we applied NPOP as sample preparation method. And when Andrew analyzed those melanoma cells, he immediately observed that there were two clusters present, and we were able to use marker genes identified by Arjun Raj at the transcript level to now color code each single cell based on the protein measurements that we had. And we were able to see that the cells from the larger cluster have higher abundance of markers associated with non-primed cells, while the smaller cells expressed at higher abundance proteins associated with priming. And there were a number of additional functional sets of proteins that showed enrichment in those clusters. And in particular, the larger cluster had enrichment for proteins associated with cell division, and the smaller cluster was associated with proteins functioning in protein secretion and so on. And whenever you do single cell measurements, there exists a possibility of inferring cell cycle information without doing any additional work. Of course, the data set has to be deep enough and accurate enough to do that. But in this case, Andrew was able to infer the fraction of cells in the phases of the cell division cycle, and that indicated that the cells in the primed cluster, in cluster B, have high probability to be in G1 phase, which is consistent with those cells being less proliferative. And furthermore, we were able to associate also molecular differences, sets of proteins that code very differently with the cell division cycle in those two different clusters. And examples of such protein sites include MAP kinase and glycolysis, that kappa B kinase cascade that appear to have different regulation across the cell division cycle in those two clusters. And there was this other really surprising thing that Andrew observed in the data. So we saw that in general, the protein content, total protein content per single cell that we measure correlates to the cell diameter. Of course, that makes sense. It's satisfying at some level to see that the efficiency of sample prep and the consistency is sufficiently high so that we can see this correlation. But we also saw that the two clusters were different, that consistently cells from cluster B had less protein content than you would expect. And you can also see this with the box plots quantified. And then we had a hypothesis that maybe those cells that are less proliferative and more quiescent-like, maybe they accumulate reserved carbohydrates, because if they don't have protein, what else can they have? RNA occupies insignificant fraction of the cell in DNA, so it's got to be something else. Maybe it's glycogen. And that harkens back to old literature and to my doctoral research, that it's a known phenomenon that slowly growing cells accumulate reserved carbohydrates. So we first looked at our data at protein levels to see whether enzyme abundances are consistent with that hypothesis. And Andrew found that they're consistent, but consistent is not the same as demonstrating the hypothesis. So then Andrew obtained more direct evidence by measuring glycogen, the main form of reserved carbohydrates in mammalian cells, and he found that indeed cells from cluster B have more glycogen, more reserved carbohydrates. And they show this example to illustrate biology that we can learn by virtue of measuring proteins in single cells, the main constituents of the cells. So it would be inaccessible if we had measured RNA in the cells. We would never have identified that. And then we could go back to the jackpot cell co-expression and look at co-expression of proteins in those cells. And from this analysis, as I already mentioned previously, we identified different functional sets of proteins. And what gave us particular confidence in those results is the fact that we could plot the results from PLEX-DOA and prioritized scope measurements on top of each other. And they very nicely lined up, showing that this gradient that we see within a cluster, which is relatively weak, and it's a kind of signal that if I only had one type of measurement, I might feel uneasy to interpret biologically. But now that we have two different methods indicating that we have this gradual change of cells becoming more primed for drug resistance and then having a sudden jump into the cluster, it's something that we can establish with higher confidence. So in the final few minutes of my presentation today, I want to switch gears and talk about probably the most important part, which is building up the community and sharing the know-how, which is what this summer school is about. It's sharing knowledge and sharing resources, algorithms, and data. So we are very keen on sharing our data and even more importantly, metadata, because if you have a lot of raw files that are not described, they're not very useful. You really need to have the metadata, and we put a lot of work and emphasis in making sure that the data can be reanalyzed and they can be accessed. All of our data can be accessed from this website. And we also worked as a community. Jorgen Cox was part of this effort in developing initial set of guidelines for performing benchmarking and reporting single-cell proteomics experiments that can be found from this website, also an article published in Nature Methods. And this initial set of guidelines is just that, initial set. So as you go about acquiring data and having new thoughts and ideas, we welcome your contributions so that we can enrich the next generation of guidelines with our experiences and with your experiences. We also have an exciting new research institute here in Boston that we are starting. As our work in single-cell proteomics attracted more attention and prominence, we were approached by prominent philanthropies, in particular Schmidt Futures, the philanthropy of Eric Schmidt, where they discussed with us the possibility of starting a nonprofit research institute. And those discussions were very favorable. So after a couple of years of writing a formal proposal and all of that, now we have this beautiful institute. That's the foyer. That's how it looks from the outside. And this is the lobby. It's about 3.5 miles from here, where we are building up the capacity to scale up single-cell proteomics. We want to increase PLEX-DOA to high plex. We are certainly not happy with having 3 or 5 labels. We think that there is a capacity to have many more. And ultimately, we really are very enthusiastic and fond of applying those technologies to big biological problems that have resisted previous efforts. So it's a resource that we would like to use to support the community. We organize workshops. So for example, we had a workshop on hands-on sample preparation a couple of weeks ago as part of the single-cell proteomics conference, and we would like to make this as a hub for the community by providing know-how and helping others learn from our experience. Everything that I told you today has been the result of the work of this team here and a few people who may not be on this picture, but it's a small team that is having a lot of fun in the adventure of developing single-cell proteomic methods, applying them to biological questions, sometimes not even single cells. They don't have to be single cells. And in particular, the NPOP method that I introduced at the beginning is developed by Andrew Leduc, who is actually here today. Gray Huffman was the lead for prioritized scope within my group. Jason Dirks developed Plex DIA, and Harrison worked on the second generation of Scope 2, and he led the project with the joint RNA and protein data that they presented. I'll stop here, acknowledging the support of funders, in particular the NIH Directors Award and the Allen Distinguished Investigator Award, and I'll be happy to answer questions.
Thank you, Nikolai, for the very nice talk. In the first strategy of pinning the proteins into different levels of priority, what were the criteria? Did you filter out the ones that are most likely to be differentially post-translation modifications, or to be like basically like was abundance of the peptides a factor?
So you can assign priority based on whatever you want. There is complete experimental freedom. So in some experiments, where we wanted to demonstrate depth of coverage and throughput, we put as high priority the most easy to identify highly abundant proteins. So that's just going deeper, going faster, having the best proteome coverage. In other experiments, we put the proteins in most of the experiments that we did based on biological relevance. We studied immune responses. We had done some bulk analysis, and we knew which are the proteins that are likely to be most biologically relevant. So we put those. Certainly, post-translationally modified proteins can go on the first priority tier. But in general, we have seen that the first tier can have 2,000 to 3,000 precursors without this having any adverse effect on data completeness. So the number of proteins that can be prioritized is quite large with that number of precursors. But the criteria for that can be anything that you desire. On the technical level, one thing that is a bit of a limitation to the extent that we're using data-dependent acquisition narrow windows, if there are some precursors that are going to be always co-isolated substantially with another precursor, that's a limitation for any DDA approach. So that's not a good precursor to put on the inclusion list. But we did not find this to be a significant limitation. So you can put there any precursor you want.
Great talk, Nikolai. Thank you so much for presenting this work. I want to ask, from a single mammalian cell, how many proteins with more than 2 unique peptides have you been able to consistently identify?
It very much depends on the mammalian cell, the method for analysis used, and the technology. So if we are to maximize that number, I think it's about 4,000 from a HeLa cell. A HeLa cell is not particularly interesting cell to analyze, right? So if we go down to analyze primary cells, that number is much lower. So that number would be 2,000 is more realistic for primary cells, that this can be quantified on relatively new instruments. With prioritized scope, you can choose a number of precursors per peptide to have, and we usually go for more than one, but not too many, so that we can quantify more proteins. But if you want a ballpark answer, that's going to be about 2,000 for most representative cells. If you just wanted to go for a large cell to report the largest number, you can probably go to 6,000. Pick a large cell, and you can do that.
That's still quite impressive. And what would be the scope of targeted proteomics, MRM or PRM, at single cell level?
That's not a strategy that appears particularly competitive to me, because of the very low number of precursors that are analyzed that way. I think that the real-time instrument control enabled by applications such as MaxQuant Live is superior to those. And in fact, you could use something like MaxQuant Live to do PRM, MRM, and achieve much higher depth. But in the more classical versions of those methods, I don't see them as being particularly competitive with what we can do with prioritized scope, where you can zoom onto a subset of 100 or 200 proteins. You can have multiple scans, maximal sensitivity, And you can still get 1,000 proteins quantified at the same time to use instrument bandwidth. To me, that's very advantageous as opposed to only have the targeting.
Impressive. Thank you. It's really interesting. I have a couple of short questions. So one is for the unpop approach. I have an intuitive concern. So do the bubble ever break to generate splash or contaminants or not?
So they're not really bubbles. So they're tiny droplets, but they don't mix. They don't splash. Yeah.
Okay, they don't change. Oh, that's great. Secondly, probably to the biological side. So if you do not use a single cell proteomics approach, probably single cell transcriptomic approach as well, do you still see alpha and beta classification for your zygote?
Not in that particular system,
No.
because the variability is simply not there. The proteins that we measure are actually not translated from messenger RNAs in these blastomeres. They're deposited there by the mother, so we don't see it.
That's great. This indicates the power of the single cell proteomics approach, of course. Then, the signature proteins, what's the fold of change, the difference between the two groups? Very small or can be large?
It is small. It is small.
Small.
So it is the larger fold changes there are about twofold.
Okay.
Maybe three, the largest, but for the most part, they're small fold changes.
But still, maybe I saw some pictures in your slides. So maybe still some proteins could be localized differently to different places or something.
We think that they are. We think that the proteins are differentially localized.
So
absolutely. We think they're differentially localized in the zygote to begin with. In fact, have evidence for that. And then as the zygote cleaves into two cells, that differential protein localization turns it into differential protein abundance between the blastomeres.
So if later then, if you do a deeper study with antibody-based staining approach or something, maybe can open, to cell biologists to a wider community, and they can follow these.
We actually did try using antibodies and other methods, and this was quite difficult to do because we wanted to validate each affinity reagent, and validating them wasn't easy. So in principle, you're right that I think that this study can open up a whole avenue for developmental biologists to delve into that. But we also found that in this case, using affinity reagents wasn't in any way easier, certainly not for us, or more sensitive as compared to mass spectrometry. Mass spectrometry was far superior than using affinity reagents.
More questions?
Hi, I'm curious if you have done any work dissociating more complex tissues for single cell proteomics, like brain?
Brain we haven't done. We have dissociated testis, we have dissociated lung, we have dissociated different tissues. So in principle, that can be done. And our strategy there is to use established protocols for doing that, the same protocols that are used with single cell RNA sequencing. And once we convert the cells into a single cell suspension, then our analysis pipeline can be applied as is without any further modifications. And one of the strengths that we found of NPOP is that it's a very gentle method. So when we work with primary cells, especially primary cells that have undergone sample dissociation, the gentle handling was very helpful, even essential to getting data. But it does work. We have applied it to cells from a number of different tissues. Brain, not yet, but in principle, if we can dissociate neurons, that should work just as well.
I just have a quick question, one that's maybe a bit naive, but I've been asked a lot for some of these plots. When you're looking at protein covariation and you see sometimes correlations that are a little bit lower in magnitude, whereas if you were to just take the raw values from 2 runs and correlate them and you get a nice 0.99 correlation, can you just elaborate a little bit on reasons for kind of differential magnitudes of correlations and different types of kind of analyses, or
So any kind of measurement noise, any kind of variation in handling samples or any batch effect is going to attenuate the correlation. So it could also potentially introduce an artifactual correlation. So if we're looking at correlations, we always have to be cognizant about the impact that measurement noise has in either inducing or weakening correlations. And if we are able to reliably estimate that extraneous variance, we can actually correct the correlation for it. And that would be my preferred approach to be able to reconcile seeing very strong correlation within a run that maybe is weaker when you look at multiple runs, that is suggesting that there's some additional technical variance that is undermining the correlation. And then if we can estimate that additional technical variance, then we can apply a Spearman correction and account for it.
Can I ask a basic question about single cell proteomics? Sorry, it's here.
Okay.
Yes, so basically about the imputations and missing values.
Yep.
So in terms of the these imputations for like a general or typical shotgun proteomics, we are expecting similarities, right? So, but for single cell proteomics, there are differences. That's why we are studying these heterogeneities. So can you comment on these strategies, how you imputed the missing values?
So with prioritized scope, missing values are much lower than with shotgun scope. If we analyze large cells from, well, larger, they're still average size, let's say HeLa cells from cell culture or pancreatic adenocarcinoma cells, we can get to over 90% data completeness with prioritized scope, the same with FlexDA. Now, as we go to smaller and smaller cells, then some precursors and peptides become below the limit of detection. So even if you have consistent analysis, you have a chance to detect it, the signal is very low, so missing value can become a problem again. So the degree to which we had data missingness varied in the various projects that they presented here. With larger single cells and with approaches such as FlexDA and prioritized scope, in some cases, missing data are relatively rare and not a big confounding factor. In some cases, there are quite a lot of missing data, and that's a confounding factor. And conceptually, that means that missing data introduces level of uncertainty. Whatever we do, however we impute it, there can be different imputation strategies. Imputation is going to make some assumptions. So the best we can do is we can know what is the generative process for that missing data, so that the imputation takes into account the generative process. For example, in shotgun data acquisition, a lot of the missingness is random because it depends whether a precursor was MS or not. Especially when using a carrier, a lot of missingness is at random. In other cases, the missingness is due to abundances being below the limit of detection. And then the imputation method has to take that into account, has to be a different imputation method. And the best we can do is just propagate this through the entire pipeline and have the uncertainty being reflected in the final result. Show that whatever conclusion we have is robust to different assumptions about data missingness and different handling of the missing data. So one approach that we have used in a number of our publications is to apply different methods for imputation or no imputation at all, simply use only the observed values for the analysis, and then see if the main result that we interpret is observed and consistent with all of the different workflows for data analysis.